Okay, so in this video we're going to see the identification test for gram-negative and gram-positive bacteria, and I'm going to perform another video about another test in general for these experiments that we're doing today, which is known as the catalase test. But for now we're going to do inoculation of bacteria in this segmented petri plates with 3 different agars. So here we have citrate agar, which is kind of dark green color, and then in here we have MSA, which is kind of light red in color. MSA stands for mannitol salt agar, and then in here we have EMB, which is eosine methylene blue agar, and this is kind of purple in color. So now another media that we're going to inoculate today is broth, and this is called phenol red broth, that it is enriched with glucose. And then inside this broth we have a little mini test tube that we call the Durham tube or Durham chamber. So within this media at least we have a source of energy for the bacteria, which is a carbohydrate, and in this case will be glucose. And whenever bacteria ferments this glucose, it's going to generate acids like lactic acid, for instance, and it will generate CO2 as part of the gases, and that CO2 can go inside into this Durham chamber, creating a bubble, and then if the bacteria produce a lot of gas, what it's going to do is going to create a big bubble that is going to make the tube to go up above the level of the edge of the media. And then for this media that we have here, MSA is selective and differential. So halophiles, or bacteria that likes to live in high concentration of salt or can tolerate it, it will grow in here. And then we have a carbohydrate, which is mannitol, that allows the differentiation of bacteria into mannitol fermenters and non-mannitol fermenters. Mannitol fermenters will use this carbohydrate and will generate acid, and it will change the color of the media from red to yellow. Similarly as for this, if the bacteria ferments glucose, it will change the color of the media from red to yellow. And then for citrate agar, in this case this media has citrate, which is a substrate, a carbon source of energy for some bacteria that has an enzyme that is called citrase. Now if the bacteria has citrase enzyme, it's going to utilize then citrate, and it's going to turn the media color from or the color of the media from green to blue. And in the case of eosine methylene blue, this is a differential media for gram-negative bacteria, so only gram-negative will grow in here. So anyway, we have then 3 bacteria to grow. So we have E. fecalis, it's here in this broth, it's E. fecalis, E. coli, and S. aureus. So then inoculation for this segmented petri plates, which is very convenient when you want to perform several tests in a single plate, and then you prevent the bacteria from going from one type of media to the other one, and you can use the same type of media as well. So not necessarily you have to have 2 different types of media. And I'm kind of giving some explanation while I'm waiting for this to cool off. But anyway, so since you have these segments that you can see here, there will be a physical barrier, like a piece of plastic, that prevents bacteria from going from one area into another one. In some cases you have a single type of media in a petri plate without segments, and then yeah, you can mark the bottom of the petri plate making the segments that we perhaps have done before. But sometimes when you are inoculating these segments without the marked segments in the Petri plate without this physical constriction for the bacteria can go from one place into another one. So, but this is very convenient. You can use these 3 types of media, or you can have single type of media, and then you can have a negative control where you don't grow anything, and then a positive control for bacteria that you know that, for instance, ferments citrate, if we have only citrate in this plate, and then negative control for one bacteria that doesn't ferment citrate. Anyway, so I think it's cool enough. So this is, let's see, this is the plate for Iphicalis. So I'm going to grow Iphicalis, which is this, into this media. And then for Iphicalis, guys, you will be kind of, since you will be doing on the 3 areas, just keep inoculating in a what we call the spot inoculation, in which you just wiggle the bacteria like this. Okay, and then close your Petri plate. Still will be just your loop, sterilize it, and then do the other segment. So the first one that I inoculated was MSA, and then I'm going to do EMB, and then citrate, and then this one. And I think that in order to prevent this video from being super long, I'm going to just do the rest of the video. But after that, and once we have the results, I will make another video showing you the results for all the 3 bacteria. Otherwise, my camera person is going to be all mad. Okay, so now it's going to be EMB, another one spot, like just side to side, and then sterilize it. Why is this? Well, this media that I'm inoculating has different components than the previous one. So what you're trying to do is prevent grabbing some of the chemicals from that media and putting it into the next media, if that makes sense. And I'm supposed to check it a little bit. And then I will do the citrate, then the phenol red rod. Okay, while I wait for this to cool off. So typically media has a pH indicator that is given by certain color, like in the case of MSA. This media is kind of neutral in pH, or concentration of hydrogen ions. And then when you have a change in pH acidity, when you increase your hydrogen levels, this media will turn from red to yellow. And there is this part in the lab report where I ask you to actually explain that process and tell more or less at what pH you have a change in the media of color, and it's in the lab report, so you shouldn't have any trouble. Now the last one that I'm going to inoculate in this plate is going to be citrate, and then sterilize, and then wait to cool off for inoculate the phenol red rod. Okay, while I wait for cool off, so enteric bacteria, which is bacteria that lives in the large intestine, some of them, they can ferment certain carbohydrates, And with this fermentation, they can produce these acids that I explained before and gas, but some of them, they don't ferment these carbohydrates and they don't produce gas. So in a bacteria that doesn't ferment these carbohydrates and don't produce gas, so you don't expect to have a change in color from red to yellow. So in this case, the media, since it's going to be alkaline or neutral, it will stay red, and then you don't see gas production. But if the bacteria ferments the carbohydrates, you will turn the media to yellow, and then you will have production of gas, but not always they produce gas. Some of them, they are capable of just using these carbohydrates without fermenting, and then they don't produce gas. So they can change the into yellow, but you don't see a bubble in this Durham chamber. So now I'm going to inoculate then this broth, just like as any other broth. You just grab your inoculum from your culture and then pass the opening of the tube and the cap twice through the flame, and then go into the middle, and then you, of course you will hit with your loop this Durham chamber. But then after that, you will sterilize your loop, and then with your tube, and your cap, pass it twice, close it, and then you're done with your aseptic technique. You clean up the area, and of course I'm going to continue with this, but this is all for now for this video. Bye bye.